Picture 20 embryos lined up on a screen. One of them has a future. A company wants to let you edit that future before it's born. In June 2026, that idea stopped being pure science fiction because scientists at Columbia University published a preprint that the gene editing process works in a lab dish. What is the story behind it? A year earlier, the company Nucleus Genomics, for thousands of dollars, promised to screen up to 20 embryos for over 2,000 traits, not just disease, height, eye color, even the odds of alcoholism. Now the company wants to go further than screening. It is funding the next phase of research inside the Columbia University lab run by geneticist Dieter Egli, who had collaborated with Nucleus's chief clinical officer, Nathan Treff, on the current breakthrough. That work showed that base editing, a CRISPR variant that nicks only one DNA strand, can correct a single faulty letter in an embryo's DNA without triggering chromosome loss. Importantly, the key base editing component was injected into the embryo as a protein, not RNA, because protein was found to be less toxic to the embryo than RNA. That's real progress. But it's not clean. The research found off-target edits and mosaic embryos where only some cells carried the fix. Egli himself admitted the technology is not ready for clinical use. The messiness doesn't stop only at the science. Treff's former company, Genomic Prediction, despite funding the current work, is suing Nucleus for misappropriating their trade secrets, a legal dispute yet to be resolved. Currently, the targeted genes are involved in regulation of cholesterol and fetal hemoglobin, which are linked to diseases and narrower than what designer baby headlines imply. But editing a human embryo for reproductive use is still prohibited in the United States and most of the world. Why draw that line so firmly? Because an edited embryo passes that genetic change to every generation after it, including any possible mistakes. Katie Hasson, who researches the social and political dimensions of genetic technology at the Center for Genetics and Society, has argued that editing embryos for reproduction is too risky to ever fully justify. Somewhere right now, a couple is sitting in a clinic looking at a screen full of embryos, wondering how much of the future they're allowed to choose. The technology in that room is real, but many of the promises surrounding enhancement are still far ahead of the evidence. Perhaps the future in front of you was never something you could fully edit. Maybe that was the point all along.